take a breakdown. This is 13.1 lecture worksheets, where we are understanding what is in every single cell in your body. If you've watched some shows where there's, crime scene investigations and they pick up a little bit of a little hair or some skin cells or even submicroscopic, you can't see hair or skin cells. You can take a swab of some saliva that's even dried up and get the DNA from that and trace that DNA back to an individual. And that's because every single cell in your body, your hair, your eyes, every single cell in your body has all of the instructions to make you. And so though it's not all expressed, it's all there, and it can link back to the original owner, your own unique DNA fingerprint. And so that's really interesting, in that's the way it's done. It seems pretty remarkable, like, wow, every single cell has to have all that instruction, but that's the way nature has evolved. It's the best way. And what it does, it just that cells have a complicated orchestra of turning things on and turning things off. And so it's best to just have all the information for everything in the cell, because it's stored so compactly. So that's what's going on. Is every cell in your body is like an atom with a center that has all the good stuff in it. And in the center, just like an atom, it's called a nucleus. Okay. And so what's in the center of a cell? The nucleus. And what does it contain? What's that good stuff? All that information. So all that information is stored on structures that are called chromosomes. Now, chromosomes are 46 very compact structures. You heard me talk about this. If you lined up all of this and it was the size of DNA, all the DNA in your human genome was the size of a ramen noodle, you'd have to basically, line it up nearly across the country. And so this is a lot of information that is stored on these very compact structures. And so it is 20 to 25,000 genes. And that's basically one gene makes one protein. One gene makes one protein. So that's what that means, is that in order to run a human, you need about 20 to 25,000 proteins. And so that's the information to make everything. And so that's proteins. Some are functional, some are structural. We'll get into the different types of proteins that are present in humans, but that to make a protein that has function, it needs the information. And that's what we're talking about with DNA and DNA on chromosomes. Okay. And so if you've heard of the 23andMe, if you've heard of 23andMe, it is a company that will sequence and give you your exact DNA sequence of your chromosomes, which is called 23andMe because there are 46 chromosomes. It's basically 23 times 2. So 23 chromosomes that are doubled. And so this is the chromosome is this shape. So let's label this is inside of here. This is the nucleus. The pink is the cell, and the center is the nucleus. Okay. So just like an atom has the center, which is called a nucleus. Inside of the nucleus of the cell are these structures, and these are called chromosomes. So this is a chromosome. It has a characteristic X shape. And so when you unwind that chromosome, you find that it is very wound up. It is wound up tightly. It's coiled, and the coils are around these proteins called histones. You don't need to know that. But when you really unwind and unpack the whole thing, what you have is this double-stranded DNA. So it's a double-stranded double helix. So here's one strand, starting here, it's one strand, and then changing colors, here's another strand. So that's what I mean by double-stranded, and we'll look at the structure of that, but that there are two strands of DNA that are linked together. It starts off looking like a ladder, and then you take that ladder and twist it around, and that's what's going on here. And when you twist it around, it's called a helix now. It's a coil. But because it's not just one coil, there are two coils. Okay, and so that's the DNA double helix. It's how information is stored as well as read. So on labeling this, let's answer some of these questions as I go along. We've got the nucleus, the chromosome, the DNA, and then if you have a section, oops, what happened to this? Okay, if you have a section, switch to pencil mode, that's what happened. Section of DNA, okay. Section of DNA is a gene. So it can be a bunch of DNA bases, okay, not just one, but maybe a run of them, 10, 100, and that codes for one protein, which is generally going to be one inherited trait, an inherited trait being like height or eye color, okay? So where the DNA is tightly coiled and packaged as a distinct structure, that's your chromosome. It's the distinct X-shaped structure, this X-shaped structure, and it's very tightly coiled within the X shape. It's not just taking the DNA double helix and making an X out of it. You can see that it's coiled up, it's coiled around these proteins, and then it's coiled up even more on itself. So it is a very tight and organized package, okay? So the areas that show this DNA double helix, here's the double helix right here, but you can also see more double helix here, and it's also in here. It's all the base structure of DNA is a double helix. When you're talking about a gene, though, this is a section of DNA, or it can be a longer section of DNA when you have a bunch of DNA bases. So here's these bases are each the individual rungs of the ladder. The individual rungs of these ladders is one DNA base. So it has to be a section of DNA, not just one. Not just one base or rung of the ladder, but a whole bunch of them. So 20, 100, and that is basically like reading letters in a sentence. So you can't just have one letter to have a word. You need to have a bunch of letters, and normally you have a bunch of letters to make a word and then a bunch of words to make a sentence. So that's what a gene is. It's a bunch of these individual bases, individual rungs on within the DNA structure, and you don't usually have 5, you don't usually have 50, maybe 100 more to have actually something that makes sense, which is the gene that codes for one protein. Okay, so this, when you have something like this, this is a genome of a human. So the human genome is going to be different from, say, like a frog genome. And we can see here, these are all of the genes that code for a single person. And the way they're represented is basically, instead of the X-shaped chromosome, which they are, it has this as all of the genes on a kind of a linear bar, and why they're black or gray. The black or gray sections are these horizontal black and gray and light gray sections. Those are genes. So those are genes, and each of these numbers, 1, 2, 3, and so on, all the way up to, you can see the XY or the XX. This is number 23, or this is number 23. Those are the 23 chromosomes in the human. There are 23 pairs of chromosomes in the human genome. Okay, and so you have this third one, or the 23rd one, is what determines your gender, and the others 1 through 22 all have the genes that code for what make you human: your height, your weight, all of the things that make your body work, your heart, your liver, your lungs, your eyes. And so that's all of that. All the majority of genetics is all very similar for us, for each of us, is that those 1 through 22 are really very similar. So each vertical bar represents one chromosome. Instead of showing it as this X, it's shown as a vertical bar with all the genes on there. So basically having a black horizontal section is a more concentrated section of genes versus a lighter gray is a less concentrated section of genes. And then there's the white in between those, the white horizontal bars. That's basically where there are no genes. So there are not necessarily the entire chromosome is a bunch of these rungs on the ladder. You know, if you take this and you coil it all up and put it on the chromosome, well, not every single rung on the ladder is information. You can see because of the white that a lot of these white spots mean that there's a lot of DNA in the human genome is not there except for just structure. It's just there structurally to connect the genes, and they're not coding for anything. Okay, so why are there two of each vertical bar? Because you get each chromosome, you get one chromosome each from each of your parents. So you get one chromosome inherited from each of your parents. And so this crosses, this combines. So here's, let's say, the chromosome from your mom. And this is the chromosome from your dad. And they combine because they are the same one chromosome, and they combine, and that's how you get the information, which then is expressed as the new individual that is kind of a mix of both, okay? And so there are a total of 23 times 2 or 46 chromosomes. And that they are 23 unique chromosomes times 2. Okay, and so this is what is different between a frog genome or an orangutan is, well, what's the same? Let's say you're talking about a frog or a tomato. Any living thing has genes. That's the information to code for that organism. Okay, whether it's a frog or a tomato or a human. And the numbers of genomes, the numbers of chromosomes in the genome might be different. The types of genes might be different. But the interesting thing is they're all made of the same exact structure, which is DNA. And so that is the setup for how genetic modification can happen, how you can take the gene from a corn and mix it with the gene from a bacteria. Is because they're all made out of the same exact thing, DNA. And so you can just take these parts and mix them together, even though they're from different species. So in genetic modification, for instance, talking about the antithrombin protein that is towards the end of that GMOs video, you could take the gene from a goat and the gene from a human, combine them together, and that becomes transgenic at that point when you're from two different species. But that is, why can you do that? It's not any magic. It's really at the base understanding that the information that codes for any organisms, it's all made out of the same parts, which is DNA. Okay, so that is why that is the setup for what allows for genetic modification. Okay. Fact that information that is inherited in any organism, whether it's a tomato or a frog or a human, is stored on the structures that are called chromosomes that are all made out of the same parts, these DNA nucleotides. So this allows for genetic modification because the nucleotides are exactly the same in a tomato or a frog or a human. And so this is the structure of one monomer, which is one DNA nucleotide. So DNA is really a polymer. So it's like a polymer that we talked about, the synthetic polymers that are plastics. The synthetic polymers are monomer. You take one monomer and you bond it over and over and over and over again. And that you can, vary the monomer, and you ended up with plastics that have different properties and different functions. Well, with biological polymers, we have already seen biological polymers in the examples of polysaccharides. So the polysaccharide, the starch, or fiber in food, it's the monomer, the one monomer of glucose, the monosaccharide. You bond over and over and over and over again. And it is something that nature does. It is so that no energy is wasted. When you just bond the same monomer, you make the same one monomer and you just bond it over and over and over again, but you vary just something slight. So in the example of the polymers that were the polysaccharides, why starch was different from fiber was how they were bonded, how the starch has the alpha bond that was the bent shape that humans can digest. And it also allows for the glucoses to get coiled up in a nice compact coiled shape. Compared to when you take the glucose and bond it over and over with the beta linkage, that beta linkage is straight and allows for the glucoses to no longer be digested, but allows for them to have strength when you bundle them together as these straight lines. So this is another example of a biological polymer. So taking a monomer bonded over and over and over again. The bonds are the same. So it's not like the polysaccharides, starch and fiber. What it is like, though, it is more like looking at the structures of the individual monomers. So it's a little bit more like the plastics that have the different functions. When you vary the individual monomer, you can vary the outcome. And that's how we end up with not all of us are exactly the same, nor are we as human organisms the same as tomatoes or frogs. That is because the individual monomers, the order of these monomers, how they're bonded together, in what order, that's what makes us all unique. So, and that's your genetic information is made out of the same parts, but not put together in the same way. So DNA is all your biochemical information to make an entire organism, whether it's a corn or a human being or a bacteria, it's all stored the same way on these structures. So DNA stands for deoxyribonucleic acid. All the parts are that you have a phosphate. So phosphate is the polyatomic ion PO4 with a -3 charge. And so this is the phosphate right here. Now the phosphate of one monomer is linked to its own sugar ring. So that's what this is, the sugar ring. And you can see it is a ring structure. It is, instead of being a straight chain, it is all connected together. So that's why it's called a ring. And the phosphate is connected to its sugar ring. And so this will connect to another phosphate of the next monomer or the next nucleotide. So a phosphate, sugar, and then the next thing would be bonded to the phosphate of the next nucleotide monomer. That would be the next bond. And then this phosphate would be bonded to the sugar of the next nucleotide monomer in that direction. And so what I referred to in the video is that DNA is a backbone consisting of phosphate, sugar, phosphate, sugar, phosphate, sugar, those phosphates and sugars coming from the individual monomers. And those are the individual nucleotide monomers, okay? And so those are bonded together, the phosphates and the sugars. What is not bonded, but what is free and hanging off the side is this part, which is the base, okay? So that's the base. And those bases are the parts that are different. So this is what varies in the 4 nucleotide monomers. So basically, when you think of what all our DNA is made out of, whether tomato or frog or human, it's these monomers, and there are really only 4 of them. It's like saying, all right, build something with only these 4 Legos, just these 4 Legos. And look at the variety that you're able to build with just those 4 Legos. It's because you can vary the order, you can vary the amount, and that leads to all the varieties we have in living organisms. Okay, so exactly how are these 4 nucleotide monomers different? So there are just 4 of these, and they are different by these bases. And so these bases are A, G, C, and T for short. And they're really actually kind of similar. So nature really doesn't try to waste too much energy. It makes things different, but still mostly from the same starting point. So cytosine and thymine both have one ring, and adenine and guanine, so let's add the A and G and the C and T. So this is adenine and guanine are also very similar. They have two rings. Okay, but they're also different in another sense, in that not all of the atoms in these rings are exactly the same. So cytosine has this nitrogen-hydrogen bond. Nitrogen and hydrogen is a polar bond. So there's a bond polarity. There's a separation of charges that's indicated by the partial positive and partial negative. And so that's for every nitrogen, hydrogen. So this is nitrogen is partial negative and hydrogen is partial positive. Okay, so adding all those partial positive hydrogen bonds in every NH bond, that's a polar bond. Okay, so this is the partial negative and the partial positive. So this would be a partial positive hydrogen here. This would be, every time you have an NH bond, you can list that hydrogen as a partial positive. Okay. And then the nitrogen is the partial negative. Let's do that in a different color. The nitrogen is the partial negative, and that's because of the bond polarity. You can calculate by the electronegativity difference, and nitrogen is more electronegative and pulls the electrons in the bond. So this is partial negative on every one of those nitrogens in an NH bond. Okay, and so what this allows for is hydrogen bonding. So remember what hydrogen bonding is it is a partial positive hydrogen that's on one molecule that then is attracted to the partial negative oxygen on a different molecule. Okay, so, and this dashed line is the attraction. This is an intermolecular force. It's between molecules. And it is really a force that is an attractive force. It is an attraction between molecules because positive and negatives attract, even partial positive and partial negative. Okay, so anytime we have the partial negative oxygen atoms, let's actually add those. The partial negative oxygen atoms that are in polar bonds, well, you have a few more possibilities. You have this carbon oxygen, that is a bond that is polar. And so that allows for a partial negative on any oxygen that has that bonded to a carbon, such as this oxygen right here, or this oxygen. Double bond, single bond, doesn't matter as long as it's carbon oxygen. So this is also a partial negative here. So having a partial negative oxygen, whether it's a double bond or single bond, allows for that partial negative that can attract to, that I did in blue, any of those hydrogens that are partial positive. Okay, now it's not just any partial positive hydrogen that is attracted to any partial negative oxygen. They have to be close together in the first place. So they have to be near in proximity in the first place. And so when you have the correct shapes that match, so this is called complementary shapes. The correct shapes, like two puzzle pieces, will fit together. Well, now the atoms are close enough together to allow for hydrogen bonding. So the complementary shapes allow for hydrogen bonding. It allows for those atoms to come close enough together so that that partial positive and partial negative can then seal the deal and keep, it's basically the glue that keeps those puzzle pieces stuck together. So when you have the exact shapes of adenine and thymine, so adenine, remember, is your A and thymine is T. And the shapes of adenine and thymine, even though adenine is very similar to guanine, well, the hydrogen bonding is not exactly the same, because look what's different. So let's show that this is an NH2 group here, right? an NH2 group here, compared to here on the guanine, it's not an NH2 group. So even though the shapes are kind of, they're the same overall shapes, but when you get down to the nitty gritty, the functional groups are not exactly the same. So the functional groups are slightly different, even though the shapes are exactly the same, which allows for specificity. And the specificity is that adenine always will bond to thymine. Okay, it will not bond to anything else and form two hydrogen bonds to link those two bases. So the two hydrogen bonds are, here's the partial positive on the hydrogen, and here's the partial negative on that oxygen. Here's a partial positive on that hydrogen, and here's a partial negative on that nitrogen. Okay, we didn't add those nitrogen partial negatives, but this is the right structures that allow for the fit and then the hydrogen bonding. There are two hydrogen bonds between adenine and thymine and guanine. Even though it is a double ring, just like adenine, you can see that, look what's different. You've got this ketone group, this carbon double bond oxygen is different. You also have a nitrogen here and a nitrogen here that are different as far as providing the partial positives that can link with the partial negatives on the matching shape, which is the cytosine. So this is a partial negative to a partial positive, and this is three hydrogen bonds that are formed that link this together. So the bond, the pairing between cytosine and guanine, C and G or G and C, is a little bit stronger because it has three hydrogen bonds compared to the pairing of A and T or T and A, that's just two hydrogen bonds. But nonetheless, the shapes allow for this, the complementary shapes are puzzle pieces that allow for these bases. These bases then are linked across the DNA helix. And that's the double helix, and that allows for two strands, two individual DNA strands to bond together. Okay, so they're bonded through these intermolecular attractions, hydrogen bonding. And you have one DNA strand with a whole bunch of bases sticking out, then you have another DNA strand with a whole bunch of bases sticking out, and they will bond together through hydrogen bonds through those bases. And so that is going back to here is actually, nope, no, going back to here. The bases stick out on one side of your DNA strand. And so it's basically like having half of a ladder. So you've got the backbone, that's the phosphate sugar backbone, and then these bases are sticking out. And then they will call for and say, hey, we need these complementary bases, and naturally form the other side of the ladder by matching up these bases, where every time you have an A, you're going to find a T. Every time you have a T, you're going to find an A to match up with it. Or every time you have a G, you're going to find a G to match up with it. And so this will naturally self-assemble when you have one strand of DNA. The other strand will naturally self-assemble by this process of complementary shapes, which we saw in the video before. Okay. Just as when I introduced organic chemistry and said, yeah, organic chemistry, it can be complicated, but when you break it down, hopefully not so bad, right? You can learn to build structures, understand what line angle drawings look like. Well, the same is true of genetics. It can sound kind of scary and you think, wow, but when you really just break it down, DNA, pretty simple. And it is something that we just need to understand. The basic structures allow for everything else. So this is on this worksheet, looking at how the DNA double helix is self-assembling. So the last thing that I had written here, when you have two strands that bond and self-assemble. So it is called a double helix. It's two coils basically, And here's one coil. Let's say you have one coil, and then you have another coil. Do this in a different color. And those two coils then get stuck together. And so that's why it is called a two-start coil, is they don't all, they're not positioned exactly overlapping, but that they're slightly off, where one start of a coil then allows for another start further up. So these two coils that are connected allow for two coils together, and the fancy name for that is called a double helix. So when you have a cell will eventually die. And what happens is, before that cell dies, it needs to make a copy of all of that information in it. And all that information is important because that's all the information to make one organism. So copying all that information doesn't take much work because of the natural structure of DNA. And so this process where a cell copies its own DNA, all of its genetic information, that process is called replication. So replication is when DNA is copied and makes an exact carbon copy, the exact same copy of the DNA. And that will be passed on to the next generation of cells. Now, just as with any information, it is important for that information to be preserved exactly as it was. So in the process of replication, there's editing to make sure that there were no errors. And it is something that is, more complicated than we need to know for Chem 9, but just know that this is happening in your body right now. All your cells are copying their DNA as needed so that they can then, die off, and the new next generation cells will be having the exact same information and not be any better or any worse. Okay, so this one strand of DNA contains all the information that's required to generate its partner strand, and that's just in the fact that the shapes are complementary. So it's driven, it's self-driven, it's self-assembled, and the self-assembly is really just A will always bond to T and G will always bond to C, or vice versa. So if you basically take a DNA strand, okay, which is two strands. So when you take those two strands and you separate those two strands, this is the beginning of replication. When you separate the two strands, you'll separate those two strands in the double helix. This is mediated by enzymes, and that will allow for the formation of copies. So when you have this strand separated, let's do a new strand in blue. Okay, this new strand in blue will have a C here and a T, and then another T will come in, and then a G and a T, and then an A. And then once those bases, the bases are called in because of the complementary base pairing, but then this backbone will bond together, also aided by enzymes, will bond together and form this new DNA double helix. Okay. Meanwhile, you'll also have the other half of it, okay, which is, I'll do a different color here. There's going to be a G that comes in here and A that comes in here. And as this builds, you will see magically, this is another new DNA double helix that forms without any overall instruction. There's no head boss, there's no brain, that because of the structures, this allows for self-assembly, and the overall result now is two copies of the original. And you can check. Is this exactly the same as the original DNA? A GC, AT, AT, CG, AT, TA? Yeah, GC, AT, AT, CG, AT, TNA. So you have now two. You have twice as much DNA, and this, in the process of replication, allows for the cell to split. So now you have two. You can have two sets of DNA. You can now split into two cells, and now you've made a copy of yourself. Okay, and that's what the cell does. each individual cell in your body that does make copies. So as long as you can separate the two DNA strands, the copies will naturally be made. And so that's kind of amazing, right? Because when let's say you want to make a copy of of this worksheet. Well, you can't just cut this in half and expect this copy, the other half, to be generated. So this is how DNA does it, though, and it's all based on the structures. So when you're given a base sequence, you can quickly write the complementary strand. And this is from the knowledge here. When you have an A, it's going to need a T. When you have a G, it's going to need a C. So let's build this. So this is going to be a T, an A, and a T, a G, an A, a C, and a G. So the second strand, the partner strand, will automatically build as long as you have this basic knowledge of A and T and C and G. Okay, so let's take a look at the next one. So when you're given this sequence, this is going to give you a C and a T, an A, a G, a G, a G, an A, and a T. And just keep on going, right? So, and then for this third one, we've got this sequence will give you a partner sequence, a complementary sequence of TAC, GGTAC. Okay, so remember the complementarity is A bonds to T and G bonds to C. And what I'm showing there are the dashed lines, which is the hydrogen bonding that that really seals the deal with those matching shapes. So genetics, not so bad, right? As long as you know that.